So thanks to all of you who are still here at the end of a day of talks, and thanks to the organizers for giving me the opportunity to talk to you today. The talks today were a lovely introduction, I think, to what I'm going to talk about and introduce the subject probably better than I could. And so since I work with biologists a lot, I have taken their custom of introducing who did the work first and last. I do it like both physicists and biologists. And so in particular, I want to highlight my excellent postdoc, Da-Peng Bi, who did a lot of the work with me that I will be talking about, some folks at Syracuse, and then some folks from the Fredberg group at Harvard as well. So I think that I don't even hardly need to show this slide anymore at this talk, but there's a lot of reasons why you might want to study cell motion in dead tissues. We had a lot of examples already this morning, but here's another wound healing movie. This is a movie of cells and the zebrafish, but now at a very early stage of development. This is the shield stage, where one tissue type is moving downward and another tissue type is moving upward, and that's sort of a type of self-organized behavior in zebrafish tissue. And you can see, and this is sort of a caricature of cancer metastasis, but the thing that all of these processes have in common is that they have cells that are very close together that are self-organizing in some way that we'd like to understand. Okay, and so the point that I want to make very first off, which I think you already saw a little bit in Pascal's talk, is that depending on where a cell is, if it's sort of surrounded by a group of its neighbors, as it is in this picture, or whether it's all by itself, those cells can move and look very different. And this is actually a slice, a confocal two-dimensional slice through its three-dimensional tissue. And what I'd like for you to notice about this movie is that you can't see it extremely well, but you can see that cells are sort of jiggling around a bit, and then every once in a while a cell will sort of change its neighbors. And so there's this very slow dynamics of neighbor change in these systems that I would like to understand better. Okay, and the sort of question that I want to propose, and it's already been mentioned by Pascal, so perhaps some features of cell motion in these tissues are a simple consequence of the cells being very crowded together. And so that's sort of a hypothesis that I want to explore in a little bit more detail and ask some sort of tough questions about that idea, because it's a nice idea. And so where does the idea come from? It comes from sort of images like this, where, again, these are the MDCK cells, and this is a picture of them sort of swirling around, and they start out not confluent, and then at some point they become confluent. And so you can see that, and then they continue to swirl, and you see patterns that look something like this. The velocity profile is very spatially heterogeneous in those systems. And so these collective swirls are sort of called dynamical heterogeneities in the glass literature, and they're sort of a characteristic thing that's associated with glassy dynamics. Another thing that's always associated with glassy dynamics, or very often, is viscoelasticity. And so each one of these things is a ball of about 10,000 cells, and it's been extracted from a zebrafish embryo. So these are three-dimensional balls of cells. And what you can see in the two different movies, this one is over seconds. So you can see it's compressed, and then it sort of springs back. Not all the way, but a large percentage of the way it sort of springs back, and that's an elastic-like behavior. And then over here you have two balls of cells that over the time period of hours are fusing together, actually precisely like two drops of oil would in water. So it really is down to the shape and everything. It really is a droplet fusion process. So the point here is that you get elastic behaviors on short time scales and fluid-like behaviors on long time scales. And that's also something that you see in glassy materials. There's been some suggestions recently that boundaries can form just due to jamming. So this is actually, it happens to be MCF10A cells now that are in a monolayer. So these are non-transformed, immortalized cells that are coming together. And what you're supposed to see here is that the cells are sort of coming together like this, and at the point where they come together, they form a boundary, or they jam here, and they don't intermix. And whether they intermix or not is actually a function of the density of the cells. And so again, whether something sort of behaves solid-like or liquid-like perhaps is dependent on some properties of how dense the cells are or what they act like. And even there's some ideas now that cell jamming is important in cancer. Of course, as Pascal showed very nicely, showed some pictures like this too, all this is saying is that depending on how confined a system is by a collagen matrix, you can either get single cells leaving a tumor, or you can get collections of cells leaving a tumor, and what sets that switch seems to be the pore size of the collagen matrix around the system. And so this is a little different, but it's still the same idea that sort of confinement can lead to differences between sort of collective type of behaviors and single type of behaviors in these systems. All right. So now that we have this idea that maybe these things are behaving in a glassy way, we'd like to try to quantify that in some precise way. So because this is sort of a school, I thought I would sort of, in case some of you haven't seen it, take you on a very quick tour of, because I come from a glassy physics background, a very quick tour of how people figure out something's a glass. Okay, how do you do that? Okay, and so the quick tour is the first thing you do if you have a glassy system is you look at the mean squared displacement. And you have already seen this a couple of times before in this school. You just look at how far something jiggles, and you look at how far it jiggles as a function of the amount of time you look at it. And if it displaces in something which is, so if the square of the displacement goes linearly in the time, then that's a diffusive process. So on a log plot, you'd want a slope of 1. And that's what you get at long time scales. And that's the time scales over which a system looks like a fluid. Okay, and over shorter time scales, a particle is trapped by a cage of its neighbor. So it sort of just jiggles around in a little cage, and it doesn't move as far as you think it should. And on that time scale, it behaves like an elastic solid. So this little plateau here is where the system behaves like an elastic solid. And this is a regime you don't ever have access to in biological systems. This is before a particle realizes that it's caged. Okay, but in biology, you usually don't have that. So what you usually look for is something like a plateau, or sort of a subdiffusive behavior followed by a diffusive behavior in the mean squared displacement. If you want to get more specific, you can actually look at individual trajectories, and people measure something which is called a non-Gaussian parameter. And that's actually pretty easy to understand. So on short time scales, you have an object which is jiggling around in its cage, and it's jiggling around in some nice sort of Gaussian way, so it looks like a Gaussian blob. And so if you said, how Gaussian is that thing? The answer would be very Gaussian. Okay? And then you let it jiggle around, and every once in a while, a rare fluctuation allows that particle to escape the cage of its neighbors, and it will jump. Okay, and so that's what I'm showing you here. There's been one jump, jiggles, jump, jiggle, jump. Okay, and after that first jump, you can see that that structure is now not looking very Gaussian, right? This thing does not look very Gaussian. So if you ask how Gaussian is that thing, you say not very. Right? And so that's a peak in this measure. So this alpha 2, the non-Gaussian parameter, is just a measure of the ratio between the fourth moment and the second moment. Basically, it just tells you how non-Gaussian something is. And so you see a peak right at this cage-breaking time scale. And then after you've jumped lots and lots of times, right, then the thing starts to look Gaussian again, and you go back down. So a peak in the non-Gaussian parameter is actually a very strong indication of this type of glassy behavior. That's sort of a gold standard, if you will. And then finally, another thing that people look at are what's called 4-point correlation functions, or chi-4, or dynamical heterogeneities. And I have to tell you, students in the room, like when I first saw this thing, I was like, holy hell, like what is that thing? It's a 4-point correlation function. So I'm going to take a second and explain what it is in simple terms. So in simple terms, the easiest thing to understand is this overlap function. And I'm going to be talking about an object Q, which is an overlap parameter, which is the sum over all of the cells or particles in the system of this overlap function. And this overlap function is very simple. It's 1 if the particle overlaps with it's still overlapping itself over a distance L, and it's 0 if it's not overlapping over a distance L. So let me show you what that means. So if I pick an L, which is 1/2 the particle diameter, that means that if the particle doesn't overlap with itself anymore, then it's 0, and if it overlaps, it's 1. So here's the particle at time t, and of course, if I look at it 0 away from t, it is exactly where it started. So this overlap parameter has to start at 1. And then after a time t plus tau 1, it's still overlapping with where it was before, and so I'm still at 1. And after another time tau 2, I'm still overlapping with where I was before, and so I'm still equal to 1. Okay, but now at a time tau 3, I am not overlapping with myself anymore, and so this function goes to 0. So this function does something like that as a function of tau. Okay? And then I average that function over all of the particles in my system. Okay? And so you can see that when a particle sort of breaks a cage is roughly when this thing is going to go to 0 if you choose this distance to be some significant fraction of a particle diameter. Okay? And so what happens then, so if I then define this overlap parameter as a function of, is a sum, okay, and now what I'm going to show you first is the average value of that quantity. What's going to happen if I choose a value which is half a particle diameter, which is this green line right here? This happens to be some data for some vibrated dense particles. This is not cells at all. But what you see is I stay at 1, and then over some characteristic time scale, I drop to 0, and this is sort of the time scale over which my system is very heterogeneous. It's where it's sort of changing. It's this sort of cage-breaking time scale. But then instead of looking at the average value of this quantity, I can actually look at the variance of that quantity. So this is what this chi4 term is that people talk about a lot in the Glassy field, and seems mysterious, but it's really not. All it's saying is I take this overlap parameter, and I ask, when is the variance in that parameter greatest? Because that's telling me when is one particle behaving very differently from another particle in this sort of cage-breaking type of way. And so you see a peak in chi4 right at some timescale tau, which tells you over which timescale is the system most heterogeneous. And it also tells you that the peak of this function tells you sort of how many particles are participating in this. So this is a really nice measure of something that you know has to be sort of varying in space and time. At the same time, this 4-point correlation function gets after it. So I just thought, in case you hadn't seen this before, it's actually not that mysterious. This is what it is. Are there any questions about those? Okay. So now, armed with these ideas about how I can tell if something is glassy or not, when something becomes completely trapped for all time so that no particle can actually diffuse at all, then I start to think of the system as being a solid, right? It's a fluid if even over a very long time scale things can move away. But so what I have here is a plot with 3 variables. It's 1 over the density, and this is the shear stress, which is applied in the system, and this is the temperature. And this is the jamming phase diagram, and basically what it says is that if I'm in this green region here, then I'm a jammed solid, which means that things can't diffuse around anymore. They're really, really stuck for good. And if I'm in this blue unjammed region, then I can continue to move around and diffuse and things like that. Okay, so this is sort of an idea that's been going around in soft matter physics for a long time. And you can actually talk about a glass transition. So a glass transition is what happens when I start out as a solid, for example, and I increase my temperature to become a liquid. That's usually called a glass transition. And then there's something called a jamming transition, which is where I sit at 0 temperature and I just change the density. And those two things have some similar features and some different features. So I just wanted to orient ourselves on a jamming axis and a glass axis before I continue. Okay, so because they're sort of, we'll talk about both of these later in this talk in relationship to tissues. Okay. One thing that you can do is you can add adhesion, because of course we think of cells as having sort of adhesive interactions between them. And so this is just a picture for adhesive colloids. So again, this is not biological systems. This is just in general what I expect to happen. And so what do you expect to happen if you add adhesion to a system of particles? The more adhesion you add, the more solid-like it becomes, right? It sort of gels up. It's sort of sticky, right? I add more adhesion. It sticks together more. I get things like gels and things like that, okay? So what this is saying is that, and it turns out actually that the right thing to do is put 1 over adhesion on this axis, and then as the adhesion increases, you get more jammed, okay? So that's just a statement of that fact, okay? So this is sort of the picture in soft matter. Okay, and now I'd like to, one last thing I wanted to point out is that if you want to prove sort of theoretically or analytically that this thing really is a transition, you can actually show that these types of transition have critical transitions. They have all the hallmarks of a second order critical phase transition with some interesting non-trivial exponents. I'm not going to talk in too much detail about this, except to say that what the fact that these data scales on two branches means. So this is the inverse viscosity. So basically, if the density is less than some critical density, then I have a finite viscosity. That's what this says. I plateau to some finite viscosity. If I have a viscosity, I'm a fluid. If my density is greater than some critical density, then I don't have a finite viscosity. My viscosity diverges, and that means I'm a solid. So this branch is a solid branch. This branch is a liquid branch, and the only thing I need for you to take away from this is that this basically shows that in a sort of solid made of sort of colloidal or granular particles, if you will, the control parameter is the area density. So I also want to take a minute and just tell you that the density here is different than the density that Pascal and a few other people have mentioned, because it's really saying what's the packing fraction of these particles. It's asking what's the space in between these particles, right? So before a tissue gets to confluence, I can change the density in this way, right? So that has to be before I get to confluence. But this is really critical for the rest of the talk. Once I become confluent, then these models say that I'm at packing fraction 1, and they don't have anything else to say about it, because they're really, everything is in terms of the gaps between particles, the actual density taken up by particles. There's a question.
Yeah, so even in this simple case, is it really in a second type phase transition? Because, for example, if you look on the bulge modulus, not shear modulus, it exhibits discontinuous.
So yes, that's actually a very good point. It has a mixed first, second order character. There's a couple of things that have a discontinuous, like the contact number jumps. So it's actually a very strange mixed first, second order transition in this case. But I think what this does make it clear is that the transition is controlled by the density. That's the control parameter. Great question. Okay, so important point, it's controlled by the density, and this is the packing fraction. These models understand a packing fraction, and they don't have much to say about what happens when you get to confluence. Okay, so what happens in dense tissue? So I just told you what we know about soft matter or glassy systems, right? So what happens in dense tissues if we look at these metrics? So these are some trajectories of three-dimensional trajectories of cells in the zebrafish embryo. And you can see that they sort of jiggle around and then jump and jiggle around and then jump. And this movie is sort of a movie of one of those moves. So this is a reconstruction, a 3D reconstruction from confocal microscopy. And you can see the cell that was highlighted with the area jiggles around for a while, and then it ducks below. So let me show you that one more time. So look at the cell with the arrow. It sort of jiggles around for a bit in a cage of neighbors, and then it just very rapidly sort of ducks out and changes its cage of neighbors. Okay, and to do this systematically, we look at the non-Gaussian parameter as a function of time. Our data for the ectoderm wasn't so good. There's some tracking errors at low time scales. So probably focus on the mesendoderm, and you see a really nice peak in the non-Gaussian parameter, and that matches up right where you see sort of a crossover from subdiffusive to diffusive behavior in the mean squared displacement. And so I would say that you really do see this sort of caging behavior in these three-dimensional tissues. Another thing that you can do is that you can look at, sorry, let me move this out of the way because I wanted to show you the movie. I thought I had this animated, but I guess I don't. So let me leave this here and move this out of the way for a second and show you movies. So these are movies of cells that are actually taken from human patients. These are primary cells. And what you can see in the movie, if I play it again, is that, oh goodness, is that you can see that there's some motion. I guess you can't see that this is much shorter than the one. Okay, never mind. You can't see any motion in these pictures. This is the cells at day 3 day 6 and day 10, and these movies aren't long enough for you to see. I apologize for that. But you can measure the mean squared displacement and calculate this chi4 variable. And so this is on day 3, this is day 6, and day 8. And what you can see is that on day 3, there's a peak at a characteristic time scale for this chi4. So this system is glassy, but it's not jammed, right? It's sort of slowing down and showing heterogeneous dynamics, but on some characteristic time scale, which is less than 150 minutes. And then on day 6, the system becomes more jammed, and you can see there starts to be a peak in this chi4, but it's off the time scale over which you can do these experiments. So this system is very nearly jammed. And then at day 8, the system is really, really jammed, and there's really no dynamical heterogeneities anymore. This thing is a solid. Okay, so you can do this. So there's really evidence, I think, that this system and the one before it are undergoing a glass transition from a liquid to a solid. So there, I think that's pretty nice evidence. Okay, so then I want to ask, well, can we explain this liquid to solid transition in models for dense tissues? That's what I'd like to know, what happens in the models. Okay, and so there's plenty of active particle models for tissues. So you saw these talks by Chate and by Hakim earlier, right, in this conference actually on Saturday. So you're familiar with these self-propelled particle models. And they have all of these sort of beautiful patterns and polarization. And if you add short-range repulsive interactions and maybe attractive interactions, if you want to mimic interactions between cells, then you recover a lot of features of tissues. That's what we saw. And in fact, there actually is a glass transition in these self-propelled particle models that actually looks nearly identical to ones that you see that I've shown you for colloidal systems or other glassy systems. So I say nearly, and that's because they have a critical packing fraction. So this phi c is the critical density at which these things jam. And what Ludovic Berthier has shown is that there is a critical density at which they jam, but it changes a teeny tiny bit as you change the persistence time for these models. So remember, in these models, there's a velocity and then there's some noise. And so the persistence time tells you how long you tend to go in a straight direction before you curve. Does that, raise your hand if that rings a bell, if that makes sense. Uh-oh. Raise your hand if that makes sense one more time, just to see how many people remember that. OK. So what this is saying is that as I increase my persistence time, it basically looks exactly like a colloidal system, except the density at which I jam changes just a tiny bit. OK? So that's the result here. But the point that I'm about to make, I want to say first, therefore, based on that sort of idea and these results, some folks, actually Fred Berg's group at Harvard, took this picture of a temperature adhesion plot. So this is the picture for colloidal solids. And they said, well, cells should just be like colloidal solids. And all I'm going to do is, because I don't have a real temperature anymore, I'm going to replace this temperature with a 1. And then I'm going to suggest this phase diagram for cells. And so this axis is 1 over adhesion. They've replaced stress with mobility here. And this axis is still 1 over the density. Okay, so all they did is propose that cells jam in exactly the same way that you see for colloidal systems. And if we believe these self-propelled particle models, you would expect the same thing. Okay, that's the idea. Okay, so unfortunately, in many tissues, this density-driven transition is impossible. It can't happen. Why can't it happen? It's because if the tissue is already confluent, then the tissue is at packing fraction 1. All of those other models tell you that something should happen at packing fraction 0.84 or something like that in 2D, right? So this cannot explain what's happening in confluent tissues, liquid to solid transitions that happen in confluent tissues. So the question is there something that can? That's what I'd like to talk about for the rest of today. And to do that, I'll talk about a model which explicitly takes into account the fact that tissues are confluent and there's no gaps between cells. So these types of models are called vertex models. They basically model a tissue as interfaces connected by nodes. And they're models generically for two-dimensional systems, sort of like epithelial layers of tissues. And there's been a lot of work on them. They've been around for about 15 years. And there's actually a lot of successes for these models in explaining cell shapes and statistical properties. All right? And so in order to write down this vertex model, you'd like to think about the forces that act to generate cell shapes. And so the forces that go into these types of models are first something like a cell-cell adhesion, which is Pascal mentioned is usually in these tissues mediated by cadherins. And then there's other associated signaling molecules with them. And so if you sort of see this outline of this double bubble, I would expect to have some adhesion molecules here. And those adhesion molecules will serve because you gain energy when you adhere to your system. They basically try to lengthen the interface. So they're trying to make this interface longer, this interface between the two cells longer. And then as you sort of heard about already, I think earlier in this school, there's also an active cortical tension. So in many of these cells, there is a sort of layer that's more dense near the edge or the cortex of the cell of actin and myosin. And it actually has the form of a surface tension. It actually scales as a surface tension. And you can actually check that Evans did by doing micropipette aspiration on cells to actually check that there is something that behaves like a surface tension, and it's way too big order of magnitude to be the cell membrane. It really is this active actin-myosin network at the edge of the cells. There's also bulk effects. For the cell types that I'll be describing, it seems to be a good approximation that the cells don't change their volume, so they resist dilation and compression. They keep approximately the same volume. That's not true for all cells, but it appears to be true for the cell types I'll try to discuss. And also you have some cortical elasticity. The cytoskeleton, in some cases, resists shear. And so we put these ingredients together, and you end up with a model that looks like this. So this basically says that if I have a cell with a given cross-sectional area, and that cell has a certain perimeter, then I can use all the ingredients I just discussed, and it turns out they come together in a really nice form. They said that the area approaches some target area with a quadratic penalty. And the perimeter approaches some preferred perimeter with a quadratic penalty. That's sort of the idea behind this model. It's actually pretty simple. And you can write that down in terms of different features. Interfacial tension, which is a mixture between adhesion and cortical tension. The 3D incompressibility plus resistance to height fluctuation gives you this area conservation term, roughly area conservation term. Okay, and so once I write something like this down, then we do what physicists do, which is non-dimensionalize it. And if I non-dimensionalize this, I say that, okay, I have two basically model parameters left. So this is the perimeter to area ratio for a single cell, and this is a model parameter. And these two model parameters, this P0 is sort of the preferred perimeter. It's actually the perimeter to area ratio, but it basically it's generated by interfacial tension, which is generated in turn by adhesive molecules and cortical tension. So that's one model parameter. And the other model parameter is this R, and you can think of it as an inverse perimeter modulus. It basically sort of tells you sort of what the fluctuations in the perimeter can be. Or you can think of it as a ratio between a bulk stiffness and a perimeter stiffness. There's a couple of different ways to think about it. But there's these two parameters in this model. Okay? So once we have these two parameters in the model, we can ask, okay, what can we do with it? And this is just a picture of a vertex model, where now what I'm doing to allow this little red cell to move around is I'm saying, well, in the absence of things like cell divisions or cell extrusion, then the only thing that can happen in order to help a cell move around is one of these T1 transitions. So this picture is a picture of the elemental moves in this system. I have two cells that are in green, and they're initially not touching. And then this there's this 4-point vertex, and then the 2 green cells stop touching and the 2 red cells start touching. And that's the only thing that can happen. And this movie here is just actually a sequence of T1 moves. So it looks like it's migrating, and it is in fact migrating, but all it is a sequence of T1 moves. And the nice thing is about these 2-dimensional systems is this is the only thing that can happen, actually. So we can calculate what's going on for a series of T1 moves and try to understand something about this system. Okay, and so what we're actually going to do, the simulation and the calculation we're going to do, is we're going to calculate how much mechanical work is required in order to execute one of these T1 transitions. So generically what we find is as we sort of shrink this edge to a point and then grow the edge in the opposite direction, we can measure how much energy work is required in the system. And you generally find a cuspy thing that looks something like this that goes up, comes back down, and then we define the energy barrier for this process, for this particular configuration of cells, to be the difference in energy from where I started and where I finished. So this is, yes?
Is it taking into account all the neighbors or only 4 cells?
It's taking into account all the neighbors. I'll show you a model where we simplified it by forgetting about the neighbors, but this calculation is taking into account everything. This transition path is simply shrinking this edge to 0 length in small steps and growing it in the opposite direction. We've tried other transition paths where you sort of create a protrusion and move it through, and a few other things we've tried. And this is generically the lowest energy transition that we found. It's not a guarantee it's the lowest one. It's hard to search all of phase space because this is a big phase space, but that's what we've chosen to be a transition path. Are there any other questions? Great. Okay. So this is the answer to your question, which is what happens if I take a very simple 4-cell case, and I can sort of, in this simple model, I'll just fix the areas of all of the cells, and I'll say, okay, I want to know what the energy barrier is. This is the energy barrier as a function of this reaction coordinate where I shrink this edge and grow the edge in the opposite direction. Okay, and this now is as a function of that control parameter P0, which again is the preferred perimeter of the cell. And so what's surprising about this is that you can say as I increase P0, the height of this energy barrier goes down. In other words, the amount of work it takes to do a rearrangement of cells goes to 0. And that's actually what this plot is showing you, is that the energy barrier actually appears to go to 0 at some fixed finite value of P0. Okay? So at this point, at least in the simple mean field model, we think that this thing is actually going from being a solid, because there's a finite energy barrier to rearrangements, to a fluid, because it doesn't cost any work or energy to rearrange. Okay? And we can actually, we sort of spent a lot of time coming up with a complicated mean field model to explain this transition point, but it's actually really stupidly simple. It's just the perimeter of a pentagon with unit area. So the point that I'm trying to make is exactly at the transition point I have pentagons, and that's the perimeter at which this system basically goes to 0 energy barrier. Yes?
Just because it seems to be important, could you say again what's P0?
P0 is the, it's actually this. So P0 is equal to the preferred perimeter of the cell divided by the cell's preferred, the square root of the cell's preferred area. And so it's just a model parameter that we can change and that presumably cells can change. I'll talk a little bit more about how a single cell could change this parameter in a little bit. It's a great question.
So maybe I mean something that if P0 is a pentagon parameter, like in configuration A, you're not at equilibrium, correct?
That's precisely right. In fact, this is sort of a non-equilibrium calculation. We're asking how much energy do you have to do to get over the energy barrier, and then it goes downhill again. That's precisely right. Okay, so the first thing you might wonder is, okay, if I'm talking about energy barriers, so this is actually, now we've done this, represents a huge amount of work because it's the energy barriers for doing every possible T1 transition in a disordered tissue and then generating lots of disordered tissues and doing it with different values of P0 and R, which are our 2 model parameters. And what this plot is showing you is that the distribution of energy barriers, so this is the distribution of energy barriers properly normalized, is universal. And it actually happens to be what's called a K-gamma distribution. That's not as important as noting that basically what this plot is saying is that it doesn't matter what my model parameters are. It doesn't matter what I make P naught or R. The distribution of energy barriers is the same. The only thing that changes is the mean energy barrier. So from now on in this talk, I'm just going to talk about the mean energy barrier and forget about the distribution because we know it's universal. The other thing which is important, and I'll come back to later, is this thing has an exponential tail. So the energy barriers in this system are exponentially distributed. So I'll mention that later too. But now we can just talk about the mean energy. So I'm going to represent the mean energy barrier in a system as overlying delta E. Okay? And so if I look at the mean energy barrier in the system as a function of P naught, the first thing you see is that they all appear to go to 0 at some value P naught, which is roughly the same as it was before for that 4-cell case, this number 3.81. And this is what happens if I change R, which is just sort of telling you about the fluctuations that you're allowing in the perimeter. So actually this looks a lot like what you see in disordered non-biological systems, where I have a control parameter at which the system goes from being a solid to being a liquid, and then there's some variable that are controlling the size of fluctuations. And so let me remind you that this sort of is like that because this viscosity controls the size of fluctuations, and then we have these variables. We have a rigidity transition which is controlled now by the distance to this critical density. Okay, so this is what you have for disordered solids, and actually this is what you have for this vertex model. So what this plot is showing you is it's showing you that this vertex model, which has been in existence for about 15 years, has hidden in it a critical phase transition with critical exponents, and it's controlled, the distance to this transition is controlled by this parameter P naught, which is this thing right here, which is just the preferred perimeter divided by the square root of the area. So this is sort of exactly like what you'd see in sort of your colloidal system or something, but now the control parameter isn't density, which is good because we see liquid to solid transitions in tissues that don't change density. They're confluent in that sense. And instead, the control parameter here is this P naught, and this P naught is controlled by things like the number of cadherins that a cell expresses, or how strong the cortical tension is in a cell. Okay, so this is something that you could imagine cells actually controlling to change a whole tissue from being liquid-like to solid-like. And as a physicist, it's also cool that it has these critical scaling exponents that we might be able to try to understand because they look kind of mean-fieldy. Okay, so theoretically, it's nice. Okay, it's also important biologically because what it says is that this picture, recall, was based on the result from particulate matter. And it says that for particulate matter, this axis should be 1 over adhesion. That is, the more adhesion I put in the system, the more solid-like I am. And that's an intuition that you get from gels, right? From make my particles more sticky and they want to stick together, right? But what this result says is that this should be opposite for confluent tissues. In other words, the axis here should be adhesion. That means that the more adhesion I have, the more fluid-like I will be. That's a little counterintuitive, but let me explain why that's true. It's because in this system, when I'm confluent, more adhesion means I want larger perimeters, right? That's what more adhesion means. And in this model, I have more perimeters, and actually that gives me more floppiness in my network. And so this model says that actually more degrees of freedom means more liquid-like when I increase the adhesion. Okay, so that's something which presumably we could test. So this first prediction is that more adhesion can actually lead to unjamming if the tissue is confluent.
Because you increase your P naught star?
It's because you increase P naught, precisely.
P naught, the transition?
No, the transition point is just a number. So P naught star is just a number. And here I'm changing where I am on this axis. So this branch where P naught is less than P naught star, I'm on the solid branch. So for all values of that, I'm solid because I have an energy barrier. So I have a finite energy barrier. And on this branch, I do not have a finite energy barrier. So this branch is the fluid-like branch of this. This is good. OK, so that's the first prediction. The second prediction is that we can also define this quantity, which is an experimental observable. So it's observable in our simulations and observable in experiments. And what it says is P naught bar is the observed perimeter to area ratio. So I actually just take a simulation or an experiment and I measure each cell's cross-sectional perimeter and cross-sectional area and I form this variable. So anytime you see something with a bar over it, this is an observable. I want to point out that that observable is different from the target perimeter. So this P naught is a model parameter. It's the target. Sometimes this observed perimeter to area ratio can be the same as the target, but it doesn't have to be, right? Sometimes the system can't go to what the model prefers. Does that make sense? That there's sort of a model parameter, which is something that's presumably fixed by cell properties. Right? And then we have this observable, and the question is the observable equal to the target we put into the model? And what you find is on the liquid-like side of the system, this number, P naught bar, exactly matches P naught. This is just a straight line of slope 1. And so what it says on the fluid-like side, the system can always achieve the target values that the model wants. On the solid side, the system cannot achieve the target values that the system wants. Okay, it's sort of, actually it's because it's stuck. Okay, but this makes a very strong prediction. What it says is, you give me your favorite model epithelial system, you figure out if it's jammed or not using some dynamical calculation, anything you want, and what we're going to predict is that if this tissue is jammed, it should have a P naught that is precisely equal to that number. Okay? Students in the audience, you may not want to try to do this because it almost never works where you theoretically predict a number for a biological system, but okay, this is what the model says. And it says as the tissue becomes more unjammed, its P naught should increase above this value. That's what the model says. Oh, it turns out that this observable also has a critical scaling. There's some hope that you might observe this in an experiment, but it would be really hard, so I don't know. This is just to show you that this observable also has a critical scaling, which is kind of fun if you're a physicist. Okay, but let's do an experiment. So this is, well, not me, but in collaboration with some really good folks at Harvard, Fred Berg's group, and these are the people that were directly responsible. We looked at these human primary bronchial epithelial cells. So again, these are cells that line your lungs, and they are ciliated. And Gina Park has come up with this really beautiful way of culturing them at a liquid-air interface and watching what they do as a function of time after they're extracted from the lungs. Okay, and so basically when we look at what's going on here, we see the following. So I'm going to show you some stuff from non-asthma patients and from asthma patients, because part of this story is that these cells are actually the cells where something pathological happens to them when a patient has asthma. There's actually a lot of debate in the literature about what precisely is different or pathological about asthma cells. And so we're interested in trying to understand that as well. So what you see is that these days go backwards. I couldn't get them to go correctly. So this is day 3, this is day 6, and this is day 8. So this is how many days these cells have been cultured at this air-liquid interface. They were taken out of a patient and then cultured at this air-liquid interface for a certain number of days. And what this color is showing you, it's the microns per minute. It's how much these cells are moving, sort of instantaneously moving per unit time. It's a measure of their velocity. And so what you can see is on day 3, the cells are very active. On day 6, they're much less active. And on day 8, they're much even less active than that. And this is for a normal patient. And you can do the usual thing where you look at the mean squared displacement and chi4. This chi4 data is actually the stuff I showed you earlier. So basically what it means is that day 3, the system is glassy, but not jammed. Okay? Day 6, this thing is glassy and not jammed, but it's almost jammed, right? Because this time scale is shifting further and further to the right. And at the longest time scales at day 8, where it looks very solid-like, this just tells you it is very solid-like. There's really no motion of cells anymore. There's no...
At this time scale, if you had to wait longer, you would see the transition, or is this diverging?
So we have not done the experiment longer than 150 minutes, partially because you start to worry about the health of these cells in this air-liquid interface after a certain amount.
Is it a semantic issue, or
No.
Only to observe yourself for 10 minutes would conclude they're solid.
Yeah, so you're precisely right. So these cells may not be jammed either. Yeah. If I were willing to wait much longer time scales, then I might find a peak at very long time scales, suggesting this system is even closer to jammed, but not quite jammed.
There's not a mathematical divergence, not that your time is diverging.
Well, so if you think that this mapping that I've just discussed is correct, where it actually does have a jamming and rigidity transition, then that model predicts that there's actually a divergence in the viscosity and a divergence in the time scale. We haven't shown anything like that here, only heuristically that it looks like it's certainly this one is much more glassy than this one. Okay, it's a good point. So the other, oh yes?
How do you know that your cells are still alive? Just stop moving because they die.
Right. So there's a couple of assays that one can do where you sort of, I forget what the name of the chemical is that you put in, and if they uptake the chemical, I don't think it's a chemical, I think it's a protein. But anyway, there's sort of a couple of assays, standard assays to check for cells still being alive. And these cells pass those assays. So they seem to be alive.
Yeah, but then why should there be any difference in behavior after a couple of days?
So that's what I will try. Can you ask me that question again in a couple of slides? Because I will try to answer it a little bit. Yeah.
Problem is that in your theory, do you assume that the thermal forces and probably some active forces in the system are much smaller than the mechanical forces?
Yes, and in fact, in this model, I'm assuming they're precisely zero. Yeah. But I will go at the end of this talk to say what happens when they're not zero.
So the phase transition is purely geometrical?
It's purely geometrical. It happens at, if you want, 0 temperature or 0 cell motility or 0 cell velocity. And we know that that can't actually be exactly correct. So I'll explain what happens when you add that back in later.
And here, which reason do you have to assume it's the same here? Because these cells have moving.
There's two answers to that question. One is to try it and see if it works. And if it does, then you ask, how come it works so well? So that's what I'm going to do. So hold your question too. It's a very good one. It's exactly the same one I had when we were doing this with this group. Okay, so the other point I just want to make for this plot is these are the asthma tissues. And you see that on day 6, so here's day 6 for the asthma patients, and here's day 6 for the non-asthmatic patients. And you can see that there's a big difference. In other words, asthmatic patients stay unjammed for much longer time scales than non-asthma patients. And this is sort of rather striking. It's a very collective effect. And you sort of wonder if perhaps some of the pathology of asthma lung properties have to do with the fact that they actually stay unjammed for much longer time scales in this assay, and I'll come back to that. But the other thing to note is that on day 6, we still see that there's a peak. So day 6 is this one here in the normal. This is day 6, I guess these squares are day 6. This is day 10, and by day 10, the system definitely does not show a peak in chi-4 over the characteristic time scale. So all I'm saying here is that the chi-4 for this system says that actually the asthma patients, all of these days are still unjammed, although they become more and more glassy again as you go down. Does that make sense? And the reason you know that they're unjammed is there's still going to be a peak in chi-4. You can sort of see there's going to be a peak in chi-4 at all those days. Okay, so the first interesting thing is that the intra-tissue tension is higher in the asthmatic tissues. So this is a picture of the average, some sort of average tension over some window in a system which is non-asthmatic. And this is the same thing in the tissue which is asthmatic. And you can see that the scale here is increasing tension. So this asthmatic tissue has twice roughly the intra sort of tissue tension as the non-asthmatic tissue. And that's just what this bar chart shows. So these are a couple of non-asthmatic systems, and this is an asthmatic system. And recall that this system is unjammed, and this system is jammed. This is taken on day 6. Okay? And so the interesting point that I want to make is that this is basically using some of the traction force microscopy that other people have showed to back out the tension in the tissue. It involves some modeling and a calculation. But the point is if I have a lot of tension in a tissue, this tissue is supporting a lot of tension, then actually that's consistent with the idea that actually the adhesion is pretty strong in that tissue, because if it wasn't pretty strong, then I would rip the tissue apart, right? So this is not, it's not saying for sure that the adhesion is higher in the unjammed system, but it's consistent with the idea that in this unjammed system, this thing is supporting a lot higher tensions in the tissue, and that's sort of at least consistent with the idea that the adhesive interactions are stronger. It's not a proof, but it's at least sort of consistent, okay? And then the other thing is that this is, again, this prediction for the, I'm sorry, the video is cutting off the bar. So this is the prediction for this perimeter to area ratio that you measure. So this is P0 bar. This is the measured quantity. Again, I'm sorry, the bar got cut off. And this is the model quantity that's just controlling the transition. And so again, the prediction is that on the unjammed side, we sort of come down and approach this value. And on the jammed side, we're at this value. So this is a prediction of a number that you should measure in an experiment. And this is the data. So this is for the asthmatic tissues, and the blue is for the non-asthmatic tissues. And in this case, because we don't know how to measure P0, but what we do know how to measure is this overlap fraction, which is this same thing that I was telling you about. It's the fraction of cells that have moved more than 15% of their diameter over the entire experiment. So basically, as this number approaches 0, we're approaching the jamming transition, right? So as we go this way, it's like going this way on this plot. Okay? And the interesting thing is that these things come down, and basically right when the overlap parameter gets to 0, so that means that the particles or the cells stay in their same spots, we're basically right at the predicted value for this perimeter to area ratio. So this is sort of suggesting that we actually see this behavior. And I think this plot shows it really nicely. So this is days 3 through 8, and we know that the system is becoming more and more jammed, and this last point seems basically consistent with jammed. And this is the same data for the asthma tissue. And basically this thing approaches this number that we predicted using this critical scaling theory. Right? So it really looks like this is telling you, in other words, that you measure the cell's perimeter and the cell's area and this epithelial layer, and you can predict just by looking at that number whether the system is jammed or not. I find that kind of crazy, but cool. Okay. So now to get back to your question, which is what's the effect of finite cell motility? Okay, and so the idea is if we look at a system and we now add some fluctuation. So basically what we've done here is we've taken this vertex model, and we've added on top of it basically the same equation that people have for cell polarization in a self-propelled or V-check type model. The same types that you've seen over and over again in this system, right, in the talks in this school. Then we can add it on top, and what you see is that if you're below P0 star, sort of you generically get some fluctuating around, jiggling around, but you stay rather solid-like. You don't really change neighbors, whereas if you're above P0 star, you do change neighbors. But we'd like to make that a little bit more precise and ask what's going on. And so what we're going to do is we're going to try to map out a line in space which on one axis is V0, which is how fast the particles are trying to move with this self-propelled particle model, right? It basically says how fast is each cell trying to move. That's V0, sort of a measure of cell motility. And on this axis is our control parameter for cell shapes. It's this P0 parameter that controls cell shapes. And what you generically find is that if you're looking at a point which is low motility and P0 lower than this transition point, you generically find something that looks like this. And we actually measure mean squared displacements in this system. And we see that the mean squared displacements don't become diffusive. In other words, this thing is really a solid. Okay? And then we go over here to a point which is sort of still below P naught, this P naught star value, but it's at a higher cell motility. And so now when we look at what happens to this system, you see that actually cells are beginning to diffuse around. In other words, this motility allows cells to move around. And then finally, if I'm here above P naught at a finite cell motility, then I get things that really can move around a lot. Okay? And so then what we could do is we can use the mean square displacement basically to write down sort of a line, and this is just using dynamics, right? To figure out where the line is between the solid-like system where things aren't diffusing and the fluid-like system where things are diffusing, right? So if you want, this is something like a glass transition line. So this is a rigidity transition like the jamming transition. And what we're finding is that there's a glass transition, there's a line of glass transitions that sort of emanate from this point, which is actually very similar to what happens in colloids, but it's actually very different. It's controlled by a different set of critical exponents, and it has sort of a different flavor. It has a different control parameter, but it sort of acts the same way. Okay, so now what we can do is we can say, okay, this was a simulation. What else can we do? So we sort of say, okay, we can sort of draw a line like this, which is sort of the line between the solid and the fluid. Okay, and now what we're going to do is we're going to look at that observable, that P0 bar, which again is the thing that you measure in an experiment, right? And that's what this color scale is. This is the thing that you measure in an experiment. Now is the function of the same two parameters. So this is cell motility, and this is the cell's perimeter to area ratio. And basically what you find is if you draw a contour line which is equal to where P0 bar equals 3.813, you see a very similar situation where if I'm above the fluid, I'm sort of above 3.813, I rapidly change P0 bar, okay? But if I'm below this variable, then P0 bar changes very little, right? So this sort of looks like maybe this is a solid region and maybe this is a fluid region. I'll come back to that. But the point is we can do two separate things. We can measure this order parameter and we can measure the diffusivity and see what they look like and how they compare. And before I sort of show you how you compare, I'm going to do one more thing, which is an analytic calculation. Yeah?
I don't really understand why you have a pentagon. So what is there so universal about the pentagon that makes it so interesting?
It's a great question. And I think the answer is that in 2 dimensions, there's something called the topological Gauss-Bonnet theorem, which says that if I have sort of a bunch of cells that are tiling space, then on average, each of them has to have 6 nearest neighbors. That's, you can prove that actually analytically. And so if you have a mean field model where you have each cell have 6 nearest neighbors like this, and then you ask, where's the transition point? What shapes of the cells? What do they look like when I reach the transition point? And remember, there's only one thing that can happen. It's just that these sort of 3-point vertices, right? This is a 3-point vertex, and this is a 3-point vertex. They have to come together and form a 4-point vertex before I can do anything else. There's actually other pathways, but we've sort of convinced ourselves that this is actually the minimal one. Okay? And so this actually, if you look at this, each one of these cells is a pentagon. Precisely at this point, each one of these cells is a pentagon.
Not the 2 on the side, the 2.
Sorry, what?
Only the top one. On the
Yeah, okay, so yes. But the thing is that actually, because we're asking what's the minimum energy required to get over the energy barrier, and so the idea here is that if you make a perimeter which is large enough to have a pentagon sit within it, it's definitely large enough to have a 7-sided figure sit within it. So you're precisely right, there's a 7-sided and a 5-sided, but you're looking for the minimum one, not the maximum perimeter. We're opposite. You're looking for the maximum perimeter and not the minimum perimeter that allows you to execute this transition. Does that answer your question? It's a very good one. Okay. So now the idea is that I'd like to try to come up with an analytic calculation for figuring out where I expect the transition from a liquid to a solid to be in the system. And the nice thing is that if I have a system that has a complex potential energy landscape. So a potential energy landscape is, as I ask, for each configuration of all of the cells, what's the total energy of that configuration of cells? And then I can sort of change all of the shapes of the cells, and that's moving around in configuration space. And this is just a 2-dimensional projection of it, but you can see as I move around in the space, I'm going to have some minima and some maxima and some minima in this space. And so what some theories of the glass transition say is that if I have a complex potential energy landscape, and I also know the distribution of energy barriers in that landscape, and I have an effective temperature in real systems, it's a real temperature, okay, but if I have a temperature and a fluctuation, basically with the statistics of that temperature, allows me to explore different minima. So I can calculate the rate at which I can, due to thermal fluctuations, that I can exit that minima and go to a new minima. It's just a Boltzmann factor with this delta E, this energy barrier in the exponent. This is an Arrhenius type of behavior. You may have seen this before. Okay? And so basically what it says is if I have a bunch of traps of depth delta E, I can calculate what the probability is of being able to jump from one to the other. And I actually really recommend this beautiful paper if you're interested in these types of models. I think this paper is incredibly beautiful if you're interested in an analytic sort of view of models for glasses. But in any case, you're trapped in a metastable state until a rare fluctuation allows you to escape. And so what we've said is, okay, maybe we'll alter this picture a little bit. We can say there's some sort of noisy fluctuations that just are like fluctuations in cell shapes, or very noisy cell motility that sort of act like effective temperature. Other people have sort of introduced that term. And maybe there's some directed cell motion that allows you just to go straight up out of a cage in a directed manner. And we're going to treat those two things differently. The details here actually don't matter that much. I can talk to individuals in detail if you're really interested in this. But basically, if you put in sort of fluctuations in motility, then these trap models allow you to write down a master equation of the kind we saw sort of earlier this morning. But now we're talking about the probability of exiting a trap. Okay, and so you can write down a master equation for the probability of exiting a trap and going to a new one. And this is sort of like, there's also some soft glassy rheology, which is sort of what happens if you add a shear stress to the system. And the effect of this cell motility is sort of like adding a shear stress to the system. Not quite, okay? But the bottom line is that this distribution, which remember came from simulations of this tissue model, has an exponential tail. And so because it has an exponential tail, that means that we know what this probability distribution, so this thing here is the probability of having a barrier of depth delta E, right? And that's exactly what we've been trying to measure, and it has an exponential tail. It turns out that an exponential tail is the only distribution that allows you to actually have a glass transition. Any other distribution won't give you a glass transition, but it turns out that if you have an exponential distribution of trap depths, then and only then you get a transition from a solid to a liquid. So this is kind of cool for us because it means that if I add cell motility to this model, I should get a transition from a solid to a liquid. And so what we do is we say, okay, we're just going to put in this exponential distribution of trap depths, turn the crank on these trap models, and we can make, this is analytic predictions actually for correlation functions in this glassy system. And then we can do some random walkers based on this correlation function, and we can get things like mean squared displacements in this system, which have a subdiffusive behavior and go to a diffusive behavior. But we can also, I think this is less interesting. What's more interesting is we can actually then make a prediction for what happens if I have a noisy cell mobility. So this is sort of what happens as I increase the cell mobility on this axis versus what happens when I change P naught on this axis. And so again, if I'm on this axis, this is what happens at zero mobility, and I go from being a liquid to a solid. But now I can also see what happens as I increase the cell mobility. And what I see is that there is, in fact, a transition from a liquid to a solid, and this line is an analytic calculation. I can also do calculations for the caging or relaxation time, and this has not been tested in an experiment, but it should be able to be tested in experiments maybe if people are interested. But we can take this and we can ask, okay, what happens then when we take all of these variables on the same plot? So this is this measure of cell motility, and this is the effective temperature that enters this analytic trap model. Okay? And what you're seeing is there's three things. There's a red line, which is sort of crummy because it's actually really hard to get good ideas of whether something is a solid or a liquid from mean squared displacement data. But the point is we basically run that simulation and see how long if something becomes diffusive or not. And so if it becomes diffusive, it's over here. If it doesn't become diffusive, it's over here, and we draw a line in between those two. And then the other thing, and this answers your question, is what happens to that observable, that P0 bar I was talking about? If I look at where P0 bar is exactly equal to 3.813, it's basically the same line. So what that tells you is P0 bar, this observable, tells you about how far away you are from the liquid to solid transition, regardless of your cell motility. Who cares what your cell motility is? This P0 bar tells you precisely how close or how far away you are from the glass transition, right? Because that's basically what it's getting the line correct. So that's kind of interesting. And that explains why the model that said, OK, we expect the transition point here at 0 cell motility actually works for all cell motilities. That's kind of nice. OK, so what it's saying is that this P0 bar is a robust metric for whether something is a liquid or a solid in epithelial layers. OK?
I mean, that comes from the calculation, I understand that. But how do you explain that cell motility don't? I mean, you would expect it, right?
Well, so as you increase cell motility, you definitely can take something which was a solid and turn it into a fluid by increasing cell motility. But the point is that all of the factors in the problem conspire to make this observable, which has to do with cell shapes, change precisely at the boundary. So let's say that you take a system which was a solid. So sitting here at a solid, and let's say you had a magic wand and could turn on cell motility. Then you would take it into a liquid, and you would see that as you go from down here, where you're still sitting at a P0 bar of 3.813, right at this transition point, you would leave it being 3.813, and then it would start increasing away from that line. So it's just saying that this observable should change, even at finite cell motilities, exactly at the glass transition point, which is kind of cool and totally surprising, and I still don't understand exactly why it's true, but it's apparently true. And also the other thing is that this analytic model, which is this green line just from this trap model, seems to work remarkably well also for predicting where this liquid to solid transition should be. And that's maybe not super surprising if you believe it and believe that there is an exponential distribution of trap depths, but it's kind of nice to see that the analytic, you sort of have an analytic explanation for why this transition is where it is in this space. So that's sort of the idea. OK, so in the last couple minutes, so basically this is sort of our big idea that actually there's this jamming transition in confluent tissues. And we think that you can explain it using some of the techniques and tools from soft matter physics. But it's very cool and different from a jamming transition you'd see in self-propelled particle models, which are governed by particle density. Here, the transition is governed by something else. So now one of the things we'd like to think about is, so far I've told you about transitions which are these T1 transitions, which involve two cells changing neighbors at a 4-point vertex. But in some systems, usually ones that have planar cell polarity or some tension anisotropy, what happens is you actually get a bunch of cells meeting at a very high order vertex and then turning to allow sort of this rearranging. And so one of the things that we are trying to understand right now is what happens when you have these higher order transitions, and how can you have them? Actually, what can lead to these types of higher order transitions? And so our model has some things to say about it, but I don't really know what they are yet. And then the other thing I wanted to mention is that I think this might be an interesting way of thinking about tumor boundaries. And the reason why is, so these are some images in a collaboration with Joseph Case's lab, where we look at, these are very malignant transformed cells. These are MCF10A, so these are basically not transformed, and these are somewhat transformed cells. But the thing I wanted you to notice is that this is just a slice through an aggregate of these cells. And what you see is that this aggregate has lots of cells that are sort of sticking out and all over and very wonky. And that is not something that you would expect from a system that is a fluid, right? Fluids will have surface tension, and they'll round up very nicely like this, which is what you see in the non-transformed cells. So the fact that you see things like this actually is a strong indication that the behavior of this tissue is not fluid-like. It's partially solid-like on some time scales. And we actually have some other data to confirm that. So the point is that tissue surface tension and ideas like that are not going to be able to tell you what's going on with these cancer cell lines because they sort of are solid-like in some time scales at least. Okay? And so the idea is that, well, maybe you could think of the EMT transition, which is this epithelial mesenchymal transition, which other people have mentioned, as basically a change in P0. Because if I have an epithelial cell, I have cells which have relatively small perimeters relative to their areas. Right? And then as I go towards the mesenchymal phenotype, I basically have these cells that are sort of much more floppy, right? And they have much bigger perimeters relative to their areas. And then the idea could be, well, the reason why it's so easy for mesenchymal cells to leave a tumor is because the epithelial cells are really solid-like, and these cells are sort of liquefying at the edges, and then they can move away. So it's a very different way of thinking about what could be generating the sort of, I mean, there's clearly many other things going on in the EMT transition as well, but it's sort of a nice way of saying, well, there's this very collective effect that could be generating this behavior in this EMT transition. So I don't know, it's an idea. So to conclude, I think that it's useful to think about biological tissues as materials with material properties that are important for biological function. Many tissue types are apparently close to a glass transition, and we get that from looking at these sort of cage-breaking behaviors and dynamical heterogeneities. Okay? And so the question is why? If they're confluent tissues, how do they undergo this transition from liquid to solid? And what I've just shown you is that this vertex model suggests that there's this novel type of rigidity transition in tissues, and that there's a control parameter, this P0, which is proportional to single cell adhesion or preferred cell perimeter. So this is sort of an answer to the question as why might cells be changing becoming more solid as a function of time? The idea would be is that it's basically some regulation of adhesion and cortical tension that is conspiring to change this P0 variable. That's the idea. And that's opposite of what you'd expect for like colloids or particulate matter. It's exactly the opposite in sign. So as you become more adhesive, you actually become more fluid-like once you're confluent. Now, there's an interesting possibility of what's called reentrant behavior, whereas you sort of, if you start out as individual cells and you come together, adhesion might help you come together. But then once you're together, the adhesion actually tends to make you more fluid-like again. So there's some interesting possibilities there. The predictions of this model are precisely realized in human bronchial epithelial cells. This perimeter to area ratio goes to exactly 3.81 in the limit that these things become jammed. Asthmatic patients have a delayed jamming transition, which we think might be part of the pathology of that disease. And I think the point I'd like to make to this audience is you never would have figured this out if you were just thinking about single cells. Right? This is clearly a collective property of many cells interacting with one another. And this could be very important biologically. I don't know if it is. It seems like it might be. And it's something that you would only have an inkling like, oh, if a cell changed its perimeter or changed its adhesion or changed its cortical tension in this way, it actually could cause the whole tissue to go from being from a liquid to a solid very, very rapidly with just a small change in parameter. And you never figure that out if you're thinking about single cells. So this is fun, and there's a lot more to do. Thank you.
Thank you very much. We have a few questions here and there.
So what do you think if you had, for example, another term like the preferred stack ratio to the energy function, would it be possible to model, for example, the pneumatics cells which I talked about, including the motion of the
so we can talk about this in a lot more detail later, but it turns out that actually the perimeter to area ratio is a metric for something like a pneumatic order. As you increase the perimeter relative to the area, you actually get a isotropic to anisotropic transition. And it turns out that you can actually model that energy functional as sort of something that has a term like a magnetization that has a Landau-like transition at 3.813. And so there's actually, I could talk to you in a lot more detail about it. The idea is that, yes, I think this model actually has possibly a lot to say about these very strongly anisotropic cell types.
Even without the additional term,
just this term does it. Okay, there's one caveat, which is that this term, well, this energy functional by itself will not give you the long length scale correlations of the time of the sort that Pascal sees. But as soon as you add a polarization to the model, in other words, the vertex model, which is just this energy functional all by itself, actually will give you a transition from sort of isotropic to elongated, but it won't be accompanied with nematic order. But if you couple that field further to a polarization field, the symmetries allow you to then have a nematic transition in the model. And so I think it will also give you a nematic transition as long as you have polarized, or maybe you don't even think you need polarized, I just think you need some sort of nematic cell motility coupled to it.
Thanks. I actually had a question regarding the modeling of the enzymatic versus the non-enzymatic tissue. I'm sorry if I didn't understand this in the first go.
No.
As the tissue crosses the jamming threshold, which parameter is being varied in the phase diagram? Is it the density of those cells?
So in our model, the thing being varied is P naught. So what that means is you can either think about it as changing the adhesion. So let me get back to that picture for you. So in our model, so this going that way means having higher adhesion, and going that way means having lower adhesion. And so the idea is that basically as you're sort of going from here to here, you're actually decreasing the adhesion going this way. But of course, we don't have access to that information in this experiment. The problem is anytime you try to mess with cadherins, you actually really strongly mess with cortical tension too, because they're strongly co-regulated. And so it's hard to come up with assays that you control P naught in a reproducible way. So all this is saying is that I don't care what's actually driving me across this transition. The point is that as I get close to jamming, which is going from 0.8 to 0 on this overlap parameter, do I go to 3.81? And the answer is yes. But we don't know what we're controlling.
So we're moving down the adhesion axis on the phase diagram.
Yes, becoming more solid. But if I could just say one more thing, it's actually, that's the way that the vertex model was derived, which was to say that this P naught variable basically decreases when I increase the adhesion. But there's another way to think about that model, and it's that cells just have a homeostatic perimeter, which is regulated in many, many complicated ways, and a homeostatic area that's regulated independently in some other number of ways. And so if you can independently regulate those two, everything goes through that I've said, because I've never told you here what this x-axis is. So we postulate that adhesion would take you that way, but actually it's not necessary for this model to work for that to be the case.
Actually, I have one more question.
Yeah.
If I may. The time dependence of the chi-4 parameter, and I'm talking the time parameter that varies on the order of days. Does that behavior of chi-4 depend on the initial conditions of the tissue? In other words, if you take this tissue out, maybe from different organisms or the same organism at different points, and you measure it, do you see the peak in chi-4 evolve with different time behaviors?
So the short answer to your question is no one's done it. It's actually very hard. This is sort of state-of-the-art chi-4 calculation. I think it would be interesting to see what my guess would be that yes, it's sort of like if you took an embryonic tissue or epithelial tissue and looked at it at different time points, that as it evolves, because embryonic tissues evolve from being more liquid-like to more solid-like, that you would see a change in this chi-4 parameter if you could measure it and do it. I mean, this is, I have to say, this chamber system is incredibly difficult to do and really a beautiful piece of experimentation that, I mean, I don't know if anyone else is really able to do these types of experiments. one other thing I'll say is that I'm sure Pascal is sitting there wondering what happens when cells divide a lot, because I haven't told you anything about cell divisions really in this thing. And so I think that this system, it happens to be in this system and a few other systems we've looked at, that cell division occurs in this system not at all, and several other systems so rarely that you can ignore it. But then there's an additional interesting question in many systems, when cells are dividing at the same time, then we haven't done that yet. It's something we'd like to talk about, but we haven't done yet.
Maybe the last question. Yeah.
In your second last slide, where you talk about the EMG,
yes.
It's very interesting to see that you propose an alternative hypothesis or a kind of way to see. Can you also comment on what future possibilities lie for clinical outcomes based on the change in, looking at it?
Yeah, so the idea that we're battering around right now is that a lot of times clinically you want to know where the edge of a tumor boundary is so that you can cut out a tumor, right? And so people have been trying, a lot of people say, well, the basement membrane that surrounds a tumor is the edge of a tumor. But quite often you get cells that break through the basement membrane but actually don't move away from the tumor. And so then a hypothesis could be that if you really want to make sure you excise the tumor, then you should be looking for, if you thought that tissue surface tension was what was keeping cells from moving, then you would try to find a mechanical or sort of signature of the edge of a group of cells that were sort of being generated by a tissue surface tension-like term. So the fact that, if this is possibly one of the things that's keeping tumor cells sort of together, right, then it sort of suggests that maybe there's a mechanical signature of a tumor boundary that you could maybe go in and look for using an AFM or some other techniques. So the idea would be is that actually if this is what's sort of generating a good effective tumor boundary, then you could sort of exploit the fact that there's actually maybe a change in this, because P0 has really got to have something to do with the mechanical properties of cells at the edge. So you might actually think that you could try to understand that.
Okay, so let's thank again Lisa.